Hey guys, decided to make a quick genetics test review. This is most of the questions. I think it doesn't have the back page on it. I don't have that on digital for my Screencast-O-Matic, but you'll get most of it. And of course, as always, we do them in class. The day before the test, you will definitely know all the answers. Number one, Gregor Mendel used pea plants to study the inheritance of traits. Remember, Gregor Mendel was the father of modern genetics. He was an Austrian monk. That was the first to notice that offspring look like their parents and investigate how that happened. He has Gregor Mendel's law of dominance that says dominant is always over recessive unless recessive is the only one present. So the answer to number one is C. Number two, he concluded that traits were inherited through the passing of factors from parent to offspring. He didn't know factors were genes and that genes had two copies called alleles, but he knew it was something being passed from parent to offspring. Number three, organisms that have two identical alleles for a trait are said to be homozygous. Homo means same, so that's two of the same traits like big T in the problem below. Heterozygous means different, and so that would be having one dominant and then one recessive, like the big T, little t in the problem below. And this is a monohybrid Punnett square. A monohybrid cross means you're looking at the inheritance of one trait at a time. In this instance, it's the height of the plant. Big T is dominant for tall and little t is recessive for short. So if you cross a heterozygous tall pea plant, big T, little t on top, with a homozygous dominant big T plant, which is also tall on the left, then you get two big T to two big T, little t genotype ratio, which gives you too tall to too short phenotype ratio. Those both can be reduced 1-to-1. So number four, in the Punnett square, all of the offspring are expected to be tall, D. The big T is dominant, and Mendel's law was dominant always shows up unless it is not present. The next problem shows what is called a dihybrid cross. A dihybrid is looking at the inheritance of two traits at the same time. Di means two. In this instance, we were looking at seed shape along with seed color at the same time. Big R is round and big Y is yellow, and those are the dominant ones over little r wrinkled and little y green. So in this Punnett square, if you cross two heterozygous pea plants for both round seed shape and yellow seed color, then you would get a Punnett square with a typical 9-to-3 to 3-to-1 phenotypic ratio. You can see in the top left corner, you get all homozygous dominant. In the bottom right corner, you get all homozygous recessive. And if you start and go diagonally from the left bottom corner to the top right corner, you see the heterozygous genotype that looks just like the parent, big R, big Y, little y. That is a pattern you will see anytime both plants are heterozygous for both traits. So you will not get this one on the test. You'll probably get a different one or be asked a different question. So you should know how to read the Punnett square, perhaps set up the Punnett square, and then either look for the genotype ratio or the phenotype ratio or answer a specific question. So for number 5, for yellow seeds, which is the dominant color, and round seed shape, which is the dominant seed shape, the answer would be 9. And you just count how many boxes have at least one big R and big Y in the same box, and that will give you 9. Number 6, when Gregor Mendel crossed a homozygous tall plant with a homozygous short plant, all of the offspring were tall because they would all get a big T allele. And so they were all tall because tall is dominant, and they all had that big dominant allele. Number 7, if a pea plant is heterozygous for round yellow peas and is crossed with a pea plant that is homozygous for round peas but heterozygous for yellow peas, how many different phenotypes are their offspring expected to show? So this one you may have to work on a scratch sheet of paper to try to figure it out. But if you just look at the gametes that these parents could make, perhaps set up two monohybrid crosses separately and just kind of look at the different ways that they could look, you can figure it out. If you see that one of the P's is homozygous big R, and it doesn't have a little r to give, there is no way to get a wrinkled P. So the answer would be they could be round and yellow, or they could be round and green. They cannot be wrinkled and yellow or wrinkled and green. So the answer would be A, only two. Situation in which one allele for a gene is not completely dominant over the other allele for that gene is called what? And we looked at this where curly hair and straight hair, if you got one allele of each, the curly hair did not take over completely. It showed an intermediate form or a blend of wavy hair. This is called B, incomplete dominance. One of the other ones we talked about was snapdragons. We did this with eye color on the Monster Lab last week. So we had several examples of incomplete dominance. It really defines itself. It's incompletely dominant. It's not completely dominant, but it's kind of dominant. 9, a cross of a black chicken, big B, with a white chicken, big W, produces all speckled offspring, or we called it the black and white checkered chicken. Both black and white feathers are shown equally. This type of inheritance is known as, this is C, codominance. Codominance means that they are both equally dominant and equally expressed. Variation in human skin color is an example of, the answer is C, polygenic traits. Anytime you have variation in skin color, height, eye color, hair color, anything where there is large amounts of variation, not just black or white, or not just tall or short, or blue or brown eyes, it is usually polygenic, which means multiple genes adding up to give that particular trait. 11, which of the following genotypes results in the same phenotype? Remember that BB is homozygous type B blood, and BO is heterozygous for type B blood. So the answer is B. Both of those genotypes would lead to the same phenotype. That's from blood type notes, and you should have that in your folder. Working a blood type problem, if a man who is heterozygous for type A blood marries a woman who has type AB blood, and they have offspring, the offspring would have what blood type? Well, if the man is heterozygous for A, he would be AO, and if the woman is type AB, she'd be AB. And so you can set up a Punnett square, and so they could have type A blood, they could have type AB blood, they could have type B blood, but they could not have type O blood. What is shown in figure 11-1? This is showing where the tetrads are lined up in prophase 1 of meiosis, and the chromatids are overlapping and exchanging genetic material. And so this is called crossing over, C, and this is what gives us great variation and great diversity. This is why you might look like your sibling, but not exactly like your sibling. 14, what are Mendel's factors called today? Mendel didn't know because they didn't know about DNA and they couldn't see chromosomes, but now we know them. They are genes. And for every gene, there's two different forms of the gene called an allele. So really for this one, A or C would work. On the test, I will just have one of those offered so that you'll know which one to pick. And I typically say gene is Mendel's factor, and then the alternative forms of the genes are called alleles. 15, the principle of dominance states that some alleles are dominant and other alleles are recessive. If it is dominant, it's always going to be expressed. Recessive alleles only are expressed when the dominant allele is not present. 16, which of the following are shown in a karyotype? If you look at them, they are paired in their homologous pairs. And so that would be there. Definitely you would be able to see homologous pairs. The first 22 pairs are called autosomal chromosomes that do not have anything to do with determining the sex. And then sex chromosomes are the 23rd pair. So in a good karyotype, you should see all of the above. The approximate probability of a human offspring will be female. It's like flipping a coin. Heads, females, tails, males. So it's a 50-50 chance. Every time you have offspring, it is a 50-50 chance. Half of the male sperm has an X and half of the male sperm has a Y, and that determines the gender of the baby. The probability that a human sperm cell will carry an X, same thing I just said, is 50% C. 19, a pedigree cannot be used to do what? You can use it to determine whether a trait is inherited. You can use it to show how a trait is passed from one generation to the next. You can use it to determine if it's dominant or recessive. So really, there's nothing there that you couldn't use it for, so D. 20, color blindness is more common in males than it is in females because color blindness is a sex-linked recessive trait. So C, the allele for color blindness, is recessive, located on the X chromosome. Females get 2 copies of X's, so they have to get it from both their mother and their father. Sons only get 1 X from their mother and the Y from their father. It is not going to be on the X and the Y. It would be on either the X or the Y. So color blindness is on the X chromosome, and so they could have 2 normal color-seeing parents, but they could get the color blindness from just a carrier female. 21, a Barr body is a condensed X chromosome that becomes inactive in the cell. Which of the following combinations of sex chromosomes represents a female? And that would be DXX. The other ones all have a Y, and anytime you see a Y as a sex chromosome, the Y makes the child male. 23, how many chromosomes are shown in a normal human karyotype? And that would be 46 total chromosomes in a normal human karyotype. We have 23 pairs. In a pedigree, a circle will represent a female. Which of the following is caused by a dominant allele? That would be A, Huntington's disease. Huntington's disease is a autosomal dominant disorder that causes the brain to deteriorate and is fatal, usually onset 30s or 40s. 26, which of the following is determined by multiple alleles? And that is B, an ABO blood group. There are 3 alleles present in the population. 27, the failure of chromosomes to separate properly during meiosis is called non-disjunction. In anaphase, for whatever reason, the chromosomes do not separate equally. If that egg has too many or too few, or that sperm has too many or too few, and that is the egg or sperm used in reproduction, then the baby would have a chromosomal problem. We looked at those in the karyotype lab. And then the last ones, we will use the figures on the following page. I don't have those with me, so starting at 28, we will pick up in class. We will finish those as well as the back of the review.